A signaling molecule, a ligand, binds to a G protein receptor and activates the G protein by exchanging GDP for GTP, leading to the activation of phospholipase C. Phospholipase C cleaves PIP2 into DAG and IP3, a second messenger. IP3 diffuses through the cytosol and binds to the IP3 gated calcium channel, opening it. Calcium ions flow out of the ER, the response. First, ligands bind to individual polypeptide receptors. The binding of the ligand activates the polypeptides, causing them to come together to form a dimer in a process known as dimerization. Dimerization activates the tyrosine kinases on the dimer by converting ATP to ADP, and thus adding a phosphate to each kinase. Specific relay proteins then bind to a specific phosphorylated tyrosine, undergoing a structural change in the process. The now activated relay proteins activate phospholipase C. Phospholipase C cleaves PIP2 into DAG and IP3, a second messenger. IP3 diffuses through the cytosol and binds to the IP3 gated calcium channel, opening it. Calcium ions flow out of the ER, the desired response.
Type 1 diabetes occurs in human cells when the pancreas does not secrete insulin, which disrupts cell communication. In a normal cell, the pancreas secretes insulin, which acts as a ligand and attaches to a receptor on the cell. This action opens up the ligand-activated ion channel, which then allows glucose to pass through the channel and into the cell to be made into ATP. However, in an affected cell, the pancreas does not secrete insulin, which results in the ion channel not opening up and the glucose cannot enter the cell to make ATP. Symptoms include extreme hunger, weight loss, and blurred vision. Treatment for type 1 diabetes is a type of synthetic insulin that can be injected into the bloodstream, which the cell will recognize and allow to bond to the receptor. This will cause the ion channel to open up for the glucose to enter the cell and produce ATP.